Welcome to section 2 of Lab Techniques. In this section, we'll be discussing blotting procedures. Let's get started. Blotting procedures are laboratory techniques that are used to identify specific molecules, including nucleic acids, proteins, and DNA binding proteins. As you can see from the slide, a southern blot is used to identify DNA. A northern blot identifies mRNA, a western blot identifies proteins, and a southwestern blot identifies DNA binding proteins. Some students keep this straight by using the mnemonic snowdrop. The S in snow represents a southern blot, and this corresponds with the D in drop for DNA. The N represents a northern blot and corresponds with the R for RNA. The W represents a western blot and corresponds with the P for proteins. This is a detailed figure of blotting procedures, which can be found in section 2 of Lab Techniques. This is specifically demonstrating the procedure for a southern blot technique. However, all of the blotting procedures are very similar. Notice that DNA is cleaved into smaller segments. Next, the DNA fragments are added to a gel and are separated by electrophoresis. The gel is then transferred to a filter, which you can see right here. A radiolabeled DNA probe, which you can see right here, is then added to the filter, which binds to any complementary strands of DNA. The filter is exposed to X-ray film and then visualized by what is known as an audio radiogram, which you can see right here. Northern and western blot procedures are similar, but in a northern blot, the gel will contain mRNA, and in a western blot, the gel will contain protein. A southwestern blot is a combination of a southern blot and a western blot. In a southwestern blot, however, proteins are added to the gel. The proteins undergo separation by electrophoresis, they're transferred to the filter, and then a radiolabeled DNA probe is added. If the DNA probe contains a protein binding region, it will bind to the protein. These protein DNA probe complexes can then be visualized when the filter is exposed to the X-ray film. Okay, let's do a question. A pathologist is studying DNA replication from E. coli samples collected from patients with urinary tract infections. What blotting technique could be used to determine if a gene is being replicated? Okay, pretty straightforward. In order to get this question right, you need to know the function of each blotting technique. Recall that a southern blot can be used to identify DNA. Therefore, DNA replication of a specific gene could be best identified by using a southern blot. So the answer to the question is B. A newborn boy is brought to the physician for genetic testing. The father has familial adenomatous polyposis, and the mother is otherwise healthy. The patient's paternal grandfather also has FAP. DNA samples are obtained from the father, mother, paternal grandfather, maternal grandfather, and the patient. Southern blotting of restriction fragments from chromosome 5 near the APC gene are shown below. What can most likely be concluded about the findings shown above? Hopefully from the question stem, you notice that the father and the paternal grandfather both have familial adenomatous polyposis. Recall from genetics that this is an autosomal dominant condition that causes the colon to be covered with adenomatous polyps. This condition progresses to cancer, so the colon must be removed before this happens. Notice that restriction fragments from chromosome 5 were used. Restriction fragments are simply fragments of DNA that are the product of DNA-degrading enzymes. Because these fragments were from chromosome 5 near the APC gene, we can deduce that the bands shown in the image must represent this region of DNA. From the image, we can see that the patient shares a fragment of DNA with the father, paternal grandfather, and the mother. Because the mother is healthy, we can conclude that this fragment right here represents a normal DNA fragment. However, we know that the father and paternal grandfather both have the disease, so that must mean that this DNA fragment right here represents an abnormal DNA fragment. In other words, we can most likely conclude that the patient has familial adenomatous polyposis.